And it's great to have so many participants in this school. It was supposed to be smaller originally. So today I am going to tell you about some of our computational work trying to understand genome organization through probabilistic models. And in this sense, I completely embrace what Ralph said, that what I cannot model, I cannot understand. But we will look at a different level of abstraction, not polymer models, but probabilistic models, which are somewhere in between. So in general, the thing I would say, if I would need to state in one sentence what we're interested in, is how is the 3D genome organization encoded or specified, and what does it do in terms of biological function? Okay, so a very oversimplified scheme would be something like this, where the DNA sequence specifies the binding of different molecules. You have kind of one-dimensional information, perhaps additional epigenetic information. This somehow specifies the 3D genome structure, and this has something to do with function. Today, I want to focus on only the following part of this flowchart, really how 1D molecular information can specify 3D genome structure, trying to understand this by modeling. And basically what I want to do is try to build a very simple step-by-step equation together with you, because it's still defined as a school. So I'll try to do it in such a way that everyone also follows and understands the details. Unfortunately, I can't see your faces, but if you have questions, you can ask at the end. By the way, we also do experimental work, but I will not talk about that today. So the data we are going to discuss is 3C data and all its derivatives, mainly high C. Of course, you heard about this set of techniques, so I don't need to say much about it. And generally, when you do high C, you get these interaction maps, where for every pair of loci, you have an amount of interactions, like in how many cells, an approximation of in how many cells do you see every two loci interacting. If you look at different scales, you see different patterns, and we want to understand how these patterns are specified. Okay, so basically we're looking for some machine, and that's the model that will connect the probability of every two loci interacting, which is kind of what we're measuring in high C. We're not really measuring the probability, but we're sampling from it. And we want to connect it to the basic way, the fundamental way that the genome specifies things, and that's through binding of molecules to the genome. Okay, so now discussing the kind of model. So mainly what you see, and this is an oversimplification, but you have kind of two extremes of models. You have the physical models, like the polymer models and so on, where you start with a model, you introduce assumptions, you introduce some mechanisms, and you see what happens. You sometimes don't need data at all for this. You might need data a little bit. There are usually a few parameters. It's easy to interpret. In terms of discovery, you can, of course, discover interesting things, as we just saw, but it's limited by essentially what you put in, right? On the other hand, you have kind of the machine learning black box models, where you start with the data, you make very few assumptions about what it is. You need a lot of data for this, but it's usually very difficult to interpret what you actually find, but you can find new things maybe. Okay, so what our approach is actually something in the middle. Okay, so the idea is to have the power of informatic machine learning models in terms of using a lot of data. We still have a significant amount of parameters, but we make it in such a way where we encode or we define explicit mechanisms that that specify what the model is, and you'll see exactly what this means in a minute. Okay, so what do we want from our model? So we want to write kind of an equation where on one side you have the probability of interaction of every pair of loci, and we want this model to be able, first of all, to explain the observed Hi-C maps. We want it to be predictive. We want it to be simple, so you don't have many features. You want it to be based on explicit biological hypotheses, to be interpretable, and then there are some technical things like you want it to be easy to calculate and technically robust and so on. Okay, so today I will now start to build this model step by step. We'll start with distance-dependent interaction, the simplest pattern, and then we'll move to TADS and genomic compartments. So let's start. Okay, so we start, the first pattern is something you saw. It's this distance-dependent interaction that when you look at Hi-C maps, when you look inside the chromosome, you see that as you move away from the diagonal, of course, the maps are very complex, but on average, you can see this trend that you have fewer interactions on average when you move away in terms of genomic distance. Okay, so now let's start together with a very, very simple model. Okay, so let's write down a probabilistic model. So a probabilistic model would look like this. So you have the probability of interaction between every pair of loci on one side. You have here the genomic distance, right, between I and J to the power of some negative number. This gives the decay. And you just need to multiply by some number to ensure that the probability is summed to 1. It's kind of a technical detail, not very interesting. Okay, and then this exactly gives you the plots that we see here. If you put them both on log axis, you see them. You see what's like a straight line here, more or less. Okay, so this is a very, very simple model, right? And the question is, what can we do with this clearly over simplified and boring model? Okay, so remember that all models are wrong, but some are useful. Okay, so before I show you and mention just what we can do, since again I wanted to give you all the details so you have an understanding of how this works, so we need to just add two tiny things. One is that we actually, let's say we have the data now, and we want to use it to fit the model. Okay, so how do we do this? What we need to add is the fact that we don't actually measure the probabilities, but we have read counts. Okay, so basically you need to put this into the following equation, which is just the probability of getting a specific Hi-C map, which is marked here by the X. Okay, so X is just the counts, basically. So it's the probability of getting a specific Hi-C map given specific interaction probabilities. Okay, so think you have the true interaction probabilities marked here by P int, and you're basically sampling from them. Okay, and in here in the P int, you can plug in whichever model you want. So we're going to start with the simple one, and then to actually find the parameters, let's say you're interested in alpha. So basically, you have all you need. This is just a function of alpha, right? You have the X, you have the I's and J's, and then all you need to do is it's just a function that you need to find the alpha that maximizes this function. This is called maximum likelihood. Okay, so this is how we fit the model and find values of parameters. Okay, so back to what we can do with this very simple model. So when I joined Job Decker's lab for a postdoc, so Job and I were talking about weird types of chromatin in weird biological systems, and he mentioned the dinoflagellates, these very fascinating species. He said, I'm interested in them, but we don't actually have a genome, so how can we do genomics? How can we do Hi-C if we don't have a genome? So he asked me if I know anything about genome assembly, and I said no, because I didn't. And I started reading about it, and I saw that it's actually, you know, kind of an open problem. So we started thinking, if we want to sequence, you know, high-C data is sequencing data, so why not try to use it in some way? And as it turns out, it was a very good idea. And I'll explain this to you in a second. I'll just mention that now there's a paper on bioRxiv with the high-C and assembled genome from Job's lab of the dinoflagellate. Okay, so basically it came from the observation that if you look at high-C in different cell types, in different stages of cell cycle, in different species, there are common features like the distance-dependent interaction. So you see that as you move away from the diagonal, you have less and less interactions. And this is in fact one of the strongest patterns in the data. In addition, you see other things like that are common, like the fact that chromosomes tend to interact with themselves more than with others on average. Okay, and this gave us the following idea, just that if you now have a set of contigs and you don't know how they are arranged, if you measure the interaction frequencies between them, now you can use this model that we learned to say, okay, if I know the probability of interaction, and it's linked this way to the genomic distance, I can just use this exact model to find now the positions of the individual contigs. And it turned out this works really, really well. And today there are dozens of genomes that are solved using high-C data, and there are companies doing it and so on. So just to summarize this part, because it's not the focus of the talk, I'll mention that in addition to de novo genome assembly that can be done, it was also published back-to-back with Jay Shender's lab. You can use this, basically the same exact idea, to detect structural variations in the genome. Now there's some very interesting work going on cancer genomes. You can use this for metagenome deconvolution, where you don't even know in your sample which contig came from which species, and you can use it for a haplotype phasing, chaining together the haplotype. Okay, so even this very, very simple model, it turns out, is useful. But now let's make it even more interesting. And we're going to move on to this pattern of topologically associating domains, or TADs. So you already saw what TADs are. Basically, if you look along genomes, you see these big triangles. Sometimes you see they're built in a hierarchical way. You see smaller triangles inside, and so on. And this was discovered in 2012. And the reason it became very interesting is because studies, for example, like this one from Stefan Munlos's lab, where the idea is that these TADs might be micro environments of regulation, of gene regulation, where promoters and enhancers mostly interact inside, although we already heard that there are some unknowns about this, and there are examples where these rules are broken. But I would say now the classical view now is, or this was, that let's say, for example, in wild type, you might have a situation where an enhancer regulates some promoter which is in the same domain, but now in a disease or a different condition, if this domain changes, now this can change the regulation, and now it changes basically the fundamental rule that you might want to interact with whoever is closest on the genome. Okay, so this is a way to break this rule, for example. Okay, so now how do we make a model of this? So the way I like to think of a model is just to define kind of what the assumptions are. It's like playing a game of what if. So what if in a single cell and a single point in time, each locus can be in one of two states. It's either insulating or non-insulating, okay? Now, loci in the insulating state, let's say they prevent interactions across them. So loci on either sides of these cannot interact. I'll show you this graphically in a minute. Three other assumptions: that the locus states are independent of each other. So if you have an insulation somewhere, it doesn't affect the state of insulation somewhere else in the same cell. Loci interact also according to distance, as we saw before. And finally, just that high C is an average over the population at a single point in time. Okay, so when we're talking about probabilities, we mean the fraction of cells in which an event is happening. Okay, so you can think about it in this way. So we have individual cells. In every locus, you could either have or not have an insulation event. And when this insulation event happens, it means that you don't have interactions going over it. Okay, and apart from that, interactions occur according to the distance, as we specified before. Okay, and now again, these are independent. You can now summarize this by just looking at the insulation probability. So how frequently you have an insulation event at every position. Okay, so let's call that C. Okay, so now how do we add that to our model? So we have the same model we had before, the probability of interaction between I and J, the genomic distance, and now we add this term. Okay, so let's see what it means. It's basically just saying it's the probability of no insulation events between I and J. Okay, so this is the probability of insulation at some position K. 1 minus is the probability of not having an insulation there. And then you multiply these probabilities for all the Ks between I and J. Okay, so this is the probability that, so for two things to interact, you need no insulation to be in between. Okay, so this is, again, very simple model. And now we want to see if it explains what we see. So again, what we do is now we take high C data, okay, and we do this maximum likelihood estimation. Basically, we find the optimal C, okay? By the way, you can actually prove that it is optimal, and there are some nice mathematical properties, but that's maybe less interesting here. And then you can, in addition, look at the map that is derived from this insulation probability and see how well these fit each other. Okay, so you're looking how well the model can explain the data that you have. Okay, and when you compare, it's actually surprisingly good in my view, because it's a very, very simple model, and you see that it's able to capture this in different species, as well as some of the more intricate details of hierarchy and so on. So just to stress how this works, so, you know, if you have this hierarchical structure, at some point people were wondering whether this might be, you know, maybe it's you have a big, this is some big loop, and within it you have smaller loops, maybe it's some very complicated structure, but this type of model says there's a much simpler explanation. You can just explain it with this insulation probability landscape. And the way it explains it is that in between the insulation, you don't have any insulation, so you just have distance decay over, right? So over here, you're going over one insulation event, and over here, you're going over two insulation events. So it's weaker because you have to multiply the probabilities, okay? So it only happens in the subset of cells where you don't have this event and you don't have this event. Okay, just a few comments. So the model has some very nice technical properties. So for example, you can take the data, downsample it very drastically, and then look at the fitted model for both the original data and the downsample data, which is very, very sparse. And you can compare how well you do this, and you see that you can go down to, you know, less than 1% of the reads and still be able to explain the data and correctly find the parameters more or less with very few reads. And this allows you to now work at higher resolution, work with less reads, which are very quite costly in Hi-C, or with less cells. You can do other things, for example, as well, just to test this model. For example, you can leave out part of the data here in gray. Okay, so here we delete once within a TAD and once between a TAD. And now you can look at the map, predicted map from the full data and predicted map from the mask data. And you see that the model is able to infer what you had. Okay, now to more biological things. So what molecules, for example, specify insulation, right? So you already heard a little bit of this, but so first of all, when we started this was initially before even loop exclusion, but we know that there are different events that, and especially now if you look in different species, that might specify what the boundaries here are, right? It could be CTCF. There are some examples of highly expressed genes and so on. Okay. In this case, it's the C. elegans X chromosome where you see these TADs. And what you see is that if you infer the insulation probability, you get very, very accurate peaks. Often it's just in a single bin with very, very high resolution, where the model tells us it thinks the insulation is located. Now, if you look at a chip of a subunit of the dosage compensation complex, you see that it very well aligns with at least some of the peaks that we find. And in this case, this was already known, but this is a way to find a very good candidate for this, okay? And in fact, when you look now at these peaks, and now you add a mutant where you knock down the dosage compensation complex, what you see is something quite interesting. You see that some insulation events are left. And as it turns out, only the insulation events that aligned with the SDC3 peaks, only these that aligned exactly between the model and the peaks were affected. Okay, so they all go away. And you see that there are other insulation events that the model recognizes, and they might suggest that there's more than one mechanism. Okay, so this is a way of dissecting very accurately what are the genomic events underlying this. Okay, another thing you want to do with the model is predict gene misregulation, or how the genome organization changes, and how that would lead to gene misregulation. So going back to this nice paper by Stefan Munlos's lab, so they have some very nice mutants where they show that what happens is in the wild type, this gene called FA4 is activated by an enhancer cluster here in purple. And in a mutant, an inversion occurs for this region. And as a result of this Wnt6 gene here in green gets activated. Okay, so here we can actually simulate it very easily with this model, because what would happen is we would just take this insulation part and flip it and then predict what would happen. Okay, and what you find is that this matches also the data. I won't show it here. It matches very nicely the mutants that they have. And this results in a disease. And you can see now that these two now are basically in their own TAD. Okay. So now I want to move on to the third pattern, which is genomic compartments. So in genomic compartments, again, just to remind you what this is it's this checkerboard-like pattern that we see in maps, okay? And the interpretation, as we saw, is that there are initially 2 states. We'll talk a little bit about the number of states and so on in a little bit, but initially that there are 2 states and each state interacts with itself. But then how do most people find what position is in what state? So you do something very weird. You use principal component analysis, and you take the first or sometimes the second eigenvector if the first doesn't work, and you do this after normalizing the data by genomic distance. So if you don't know what this does, I'll just say you can think of it as it's some method that will try to uncover what these are, but it's basically data analysis. It's not a model. Okay, so it's some aggregation. I won't explain exactly what this does, but basically it has many caveats, and it comes from the fact that it's not a real model, because what you would want to do is now take this story about 2 states that interact with themselves and write it down as an explicit model. And the PCA has many caveats. It breaks down at high resolutions. It's difficult to interpret what the values exactly mean and so on. Okay, so again, let's play this game of what if. So in a single cell and single point in time, let's now assume that each locus is in one of 2 states, A or B. So if you're not A, you're in B. Loci of the same state can interact, and locus states are independent of each other as before. Loci interact according to distance as before, and as before, high C is an average over the population at a single point. Okay, so now we just have states, and states interact with themselves. Okay, so this is basically what we think is happening. This part was led by Tal Feld and Hagai Kariti from my lab. And let's think about it in this way. So in every cell in the population, every position can be in one of 2 states. So we write it here as A or not A, which is essentially B. Okay. Again, we can summarize this as this landscape. We'll call it lambda, which is just the probability of every locus being in A. Okay, so it's the frequency again in the population. Okay, how do we write it in the model? So we again start with the distance decay. Now we remove the TAD part. You can ask me later about combining those if you like. And now we essentially just write what I said in words previously. Okay, so things, in order to interact, both I and J need to be in A, or that's plus in probability terms, they both are not in A or in B. Okay, so that's it, very simple. Okay, you plug it into the what I showed you before, and now you can estimate, for example, the parameters. Okay, so now given high C data, we calculate for every position this lambda, the fraction of cells in which it is in state A or B, which is the opposite. Okay. Now we compare the data to predict the interaction map as we saw before. You can see the data up here and the model down here. You can see by eye, it explains quite well. It doesn't explain everything. For example, one interesting thing that it doesn't explain is the TADs. Okay, you can see it quite clearly here by eye, I hope. So you need something separate to explain TADs. And I think this is, for example, a nice demonstration that TADs are distinct from genomic compartments. Okay, so it fits well. It fits better than PCA, in fact. It works well in high resolutions. Again, there are some nice technical properties. So for example, you can do the same as we did before. You can take high C data, calculate the lambdas from it, and then downsample and calculate the lambdas and see how off you are by downsampling. And again, you can go to very small fractions of a % or less, and you get a very small error. Remember that the lambdas go between 0 and 1. That's the probability of it being in a state. Okay, so this is up here, it's still a small error. Okay, and you can go to very high resolutions like 5 kb with very few roots. Okay, in addition, to verify that this is makes sense biologically, it's known as you know, that these are correlated with histone modifications and chromatin activity. And indeed, we find that the model is better correlated in terms of absolute Spearman correlation with these histone modifications. Also, you can look, for example, at the locus state distribution. If you look at the PCA eigenvectors, you get kind of a weird shape. If you look in the model, now this actually has a meaning. If you put a division here, it means this is exactly the point in which half the cells are in A and half in B, and you get a nice bimodal distribution. So there's a very nice behavior as well. But now look at this. So how would a two-state model explain this? So you start thinking about it, you see it can't. Okay, so you know there's A and B, but now look at this. Look, if you look at the bottom, right? This is, you know, this part is very dark, and these are light. Now this one is dark, light, dark, light. The one above it is opposite. Then you have all kind of weird things going on here. You can quickly conclude that this is, you know, that our model is oversimplified. Okay, but what is nice here is now we can use this framework to make it easily extended to more states and so on. So let's see how we do this. So you can just think of the model we wrote this previously. You can just rewrite it in a very simple way. 1 times the probability both are in A, 1 times the probability both are in B, 0 times the probability this one is in A and this one is in B, and 0 times the opposite. So you can think of it as a matrix. Actually, if you write down the math, it also works with matrix multiplication. But so you can think of our model currently as this, and now you can extend it. Okay, so you can ask, now let's extend it to a bigger model. Are there more than 2 states? Do states self-interact with similar strength? Okay, so for example, should these be 1? Are they different from one another? What about the zeros off diagonal? Perhaps states interact with each other in different ways. Okay, so we can now use this framework to discover these things. Okay, so now I want to show you results from what happens when we try a 4-state model. Okay, when we try a 4-state model, we get something which was to me very surprising. So we get a matrix, first of all, which is diagonal. So we get zeros off diagonal. To me, this was quite surprising. I didn't expect it. And we get numbers here that are not equal to each other. So first of all, there are more than 2 states. I will show this in a minute. I will convince you, and if the previous one didn't convince you, I'll show you that this model makes sense. Second, the states self-interact with different strengths. And finally, the states, it seems from this, do not interact differentially with other states. So they all have these 0 off diagonals. So how do we get these complicated patterns? I'm almost done. So what we see is something very interesting, which is state mixing. And we see this, it's quite a common feature of chromatin. So look at these, okay? If you look at this bottom part, and you call this state 1, and the middle part, state 2, now notice if you look at the top strip, it looks like a combination of both, okay? So what we identify is that there are regions which are not if you are familiar with, there's something called subcompartments, which said, okay, you know, this is a different pattern. Let's say it's a different type of state, but this is not a different state. It's a mix between the states, okay? So this is a very interesting feature that we find is quite common, in addition to the fact that we have multiple states very clearly according to this model. Okay, so I'll just summarize by saying that we, again, understand, and understanding is modeling 3D genome organization with these explicit probabilistic models. We start with this very simple model, which can be used for genome assembly, and then we complicate it a little bit to add TADs, get TADs by insulation, and genomic compartments by adding states. So I'll end with this and thank my lab members. Part of this was in collaboration with Yob, and that's it. Feel free to ask questions.
Thank you very much, Noam, for these great talks. Yes, we have a question from Angelo. Angelo?
Yes. Hello, Noam. How are you?
Hi.
Thank you very much. Sorry, just a curiosity. Can you also study mutation? I mean, with your so
Yes.
if you I don't know, just imagine if you change if you make small modification in your once you have learned this, for instance, these C parameters, can you, if you just
so
change
structural,
them,
so structural variation is very easy to simulate. For example, something like a deletion, you just take the insulation and delete it. And you can do the same also in the compartment model. And actually, it's quite interesting to think of what a deletion does in the compartment model, in the state model, and in the TAD model. And now we're working on combining these. And it's very interesting because they can have very different effects on how things work. So definitely for structural variations, we can also simulate things like, if there was a global reduction in the amount of insulation or something like that. But this is not sequence, so you need to assume if you make a change in sequence, like a SNP or something like that, you need to make an assumption about what that does to the insulation or to the state or something.
Okay, thanks.
Yeah.
Okay, there is a question from Edlem Aljibouri. Have you considered adapting your model for prediction of the insulation boundaries to include the loading probability?
Yeah, so it's very interesting to consider how to modify this in terms of loop extrusion. So yes, we're definitely thinking about this. We haven't really done anything yet, but I think things like the directionality that you have from CTCF, of course, the models I show you, for example, can't produce things like small localized peaks, the focal point interactions. That's something that these models can't produce. So that's another component that definitely we need to add if we want to, at some point, reach a complete model.
Another questions from Ilya Fiammer. Your compartment model for two states sounds very similar to C-score. I don't know what is C-score. Is there any difference? And the question is, okay, are your models available to try? So you can discuss this in the Slack channel. But first question is the comparison with the C-score.
I'm not sure what it means by C-score, but I'd be happy to discuss it.
Okay.
Yeah.
Okay, so another question, is this statistical model able to reproduce single cell data?
Yes, this is very interesting. We haven't tried it yet. This is the short question, but we're definitely starting to look at it. You need to make some modifications here to account for the fact that in a single cell you can sample every fragment only one time at most. This is something which is currently not in this model, so that's something you would need to add if you want to accurately do this. But yes, this is another very interesting direction.
Okay, so question from Daniel. Daniel?
Yes, hi Noam, thanks for the talk. I was wondering, in the part where you, for the TAT part, where you infer some insulation score, did you compare this insulation scores to, I don't know, CTCF or cohesing ChIP-seq to see how they correspond?
Yes, so actually we've done something even better, I think. So what you can do is you can take a track, let's say CTCF track, and say, what if this was the insulation? Let's use this as if this was the insulation, maybe add some parameters that would allow to scale it in some way, and try to directly fit this to the data. And what you see is that it explains only part of the places. So there are clearly places where there are mismatches. So there's in both directions. So you can have CTCF and no boundary, and you can have a boundary without CTCF.
Okay, and
but I think
rapid second question for you is the last part on the AB compartment. So you put some kind of effective interactions between this compartment. Can you account for cooperativity? Because I think we know that, I mean, For example, the folding of this in this block copolymer model, for example, that we are using, this folding is highly cooperative, meaning it depends on the size of the blocks or on the local arrangement of these blocks. So are you accounting for that also in your model, or is just very simple, like direct?
Yeah, so no, so if I understand, you mean cooperativity, would it add dependence between the positions?
Yeah, it's more like, yes, that the interactions between two beans depends on the surroundings somehow, not only on the state of being.
So we assume them right now to be independent, but it would, this is another very interesting direction to add some kind of dependency between the positions.
Okay, thank you.
Yeah.
Okay, another question from Thomas Sabate. In the paper of Rao et al. In IC on coenzyme diagram, where they remove the coenzyme, they define 5 or 6 epigenetic subcompartments. Did you try to use more than 4 different states for your model? And why did you choose 4 different states?
Yeah, so a good question. So basically the way they found the compartments there, well, the subcompartments, was basically by clustering, okay? So for example, if you look at their compartment annotation, in this example, for example, they say these are 3 different compartments, okay? But what we think is that this is not a fundamentally different state, but it's actually a mix of states. But we're actually exploring if this is something different biologically, it has a different biological meaning. So the question of how many states there are, it's actually not that easy to answer, but we think that there are at least 4. Currently, you can add more states. You have the problem that you usually have with models, that when you add more parameters than usually you do, at least as well as the previous part. So I think we won't have a good answer for this until we combine the model with the TAD model. Because what happens is that when you add too many states, it starts trying to explain TADs. So I think you need to explicitly add the TADs to the model, and maybe then we'll have a better state. But I would say we're pretty confident that you have at least 4 states.
And I have a question about this. Can you allow to have repulsion between states, not only self-attraction?
Sorry, can you add?
Can you allow to have repulsion?
Yeah, it's possible, but we haven't tried it.
Maybe last question from Vera. Vera?
Yes, hello. So actually, I've seen that other people are asking them similar questions. So this is very interesting, and it just brings the question of how this relates to the states that we can define with epigenomic marks. You know, have you tried looking? So this is the first question. And then I have just another really short one, which is, do you find, because you said that basically different states attract each other, well, they have different affinity for each other, right? Yes. So do you think that the repressive or the more open chromatin dominates the, the structure? But you can start from the first question if you want.
Okay. But let me start from the second actually. What we found is that they have, at least from the current picture, it seems like they have different self-interaction, not different interactions with each other. With each other, they interact in a similar way. This is at least what our current findings suggest.
But your numbers in the diagonal were slightly different, right? There was some 0.20 something, 0.4.
Yeah, so that's the self-interaction, each state, how it interacts with itself.
Yeah, that's what I mean.
Yeah, so I'm careful a little bit of how to interpret this, because it first seemed that it had a link to some that with activity and so on, active state versus non-active state. But now we're actually using this also on microscopy data, and we're getting slightly different numbers. So I'm not 100% sure about that. So I don't want to make any claims about the direction it goes, but I think it does seem that there are differences. Now, regarding the underlying epigenetics, of course, this is very interesting. What was surprising to me is that when you read current literature, it seems, and I think this is one of the reasons why people are maybe slightly less interested many times in compartments. It seems like, oh, everything is explained. It's just active and non-active chromatin, right? That's what kind of everyone says. If you try to explain it with a model, I thought, of course, it would work. Actually, see, it's not easy at all for a model to explain this. And we're currently exploring. To me, it's actually surprising how difficult it is to find a rule that explains these states using the underlying histone marks.
Right. Yeah. So maybe, I mean, the problem is that obviously we have defined, there are models with 11 different chromatin states, and you just wonder, is it really necessary? But certainly we cannot be too simplistic. I think 2 states is really just not enough. So you have to imagine, constitutive heterochromatin and facultative heterochromatin, plus different states of the active chromatin. But also there might be a problem with species, because I think if we want general principles, we will hit some differences. Some species have methylation, some don't. And I think there might be small differences that just prevent the identification of universal principles. But I still think that it's still necessary also to combine epigenomic data and what you mentioned, the microscopy. I think it's fundamental, because sometimes I wonder, all the knowledge that we can extract from sequencing-based methods. Sometimes I fear for everything we do not see because of the technique and not because it's not there. So
yeah. I agree completely, and I think, by the way, I think this is one. So we tried this with many tracks of histone marks, especially in GM 12878, where you have lots of tracks, and you clearly see examples where you have the same state and different kind of histone marks, and also different histone marks with the same state. So it's not clear to me what's going on.